catch the fish, the first thing you would do is you would enter it into your handheld GPS unit, and it would keep track of latitude and longitude of where you caught that fish. And down at the bottom, if it was a female or male, there's eggs, milt, or uncertain little areas you would mark on there. I'm going to show you how we do the DNA and scale recovery on the ocean with, of course, a stuffed fish. When the fish comes out of the water, he lays down, and after you've killed him, or if it's catch and release, you would have him laying in a net very gently. You take your packet, little pouch that has a piece of paper and a barcode tag in it, corresponding to the barcode on the envelope. And you would take a piece of paper, you open it up. On this part of the fish, you would take a few samples out of there, either with the back of a knife you can rub, or with tweezers. You get about 8 or 10 scales, and you would smear them in the paper, and then you would fold that over. And you would take scissors and you cut just the very tip off of that fin right there for the DNA sample, and you smear that down in there, and you fold that over, and you put it back in your pouch, and you take the tag and you would put a zip tie through the lip and hook that on for future purposes. And then you take your envelope and you would write your vessel name, date and time, depth of capture, fork length. You measure from the tip of the nose back to the fork of the tail. And then there are some marks here. You look over the fish, and different hatcheries do different markings when they release fish. Some have the adipose fin clipped. You would mark that on here if this was gone. Some have some fins missing. Sometimes they'll put a dye spot right here. There'll be a yellowish circle. It's just a dye mark. You would mark that off. Next thing you would mark, you took scales, you took DNA. Occasionally, we will take stomach samples and put them in a bag, and you would mark that. When you get in from fishing, you would have hopefully a whole wad of envelopes that you would give to your port liaison, who would then in turn take and give to the scientists to do their work. You would give it to Renee to do the DNA.
Here I have a packet of samples that were taken by the fishermen. So in every single packet is a DNA tissue sample and scale sample for a single fish. Here in the laboratory, we open up each packet and we take a very small piece of the fin clip and use that for our DNA extraction.
Microsatellites are the key to matching genetic identities. CSI TV shows use microsatellites to match a small sample of crime scene DNA to a criminal. Microsatellites are used to determine paternity, and microsatellites are used to match ocean salmon to their rivers of origin, thus identifying whether they are of weak or strong stock. To understand microsatellites, you need to understand genes and their alleles. And to understand genes, you need to understand DNA. For all life forms, the DNA code comes down to the sequencing of 4 nucleotides. These nucleotides are paired together in the middle of the double helix and look like rungs of a ladder. The 4 nucleotides are adenine, A, thymine, T, cytosine, C, and guanine, G.
So what's a gene? A gene is simply a sequence of nucleotides in the DNA double helix. It might code for a protein, or it might have a different function in the genome, such as a regulatory element. A gene has a consistent location on the chromosome for individuals of the same species. This is called a locus. If you have variant copies of a gene, these are called alleles. So for example, everybody has eyes. We have genes that code for colors of eyes, but one person has brown eyes and one person has blue eyes. These are examples of alleles or differences that are encoded by genes. A gene is considered a basic unit of heredity because it codes for a protein. So what's a microsatellite? A microsatellite is like a gene, only it doesn't code for a protein in most cases. A microsatellite is a string of nucleotides that are repeated numerous times in the DNA code. For example, CA, 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 CA. That's a microsatellite. The number of repeats can vary between individuals of the same species. As with the gene, when different individuals of the same species have different versions of a microsatellite, these are called alleles. However, unlike genes, microsatellites do not usually code for proteins, which means that they do not influence the characteristics of an organism. If a microsatellite does not code for a protein, a mutated allele is more likely to be retained in the population because it's not under selective pressure. Another way of saying this is that mutations in genes that code for proteins are usually harmful. And because they're harmful to the individual, that individual is selected against in the population. And they typically don't survive to reproduce and pass their genetic material on to the next generation. But if you have a microsatellite mutation, which doesn't influence the fitness of an individual, that mutation can stay in the population because that individual is not being selected against. Because of that, you can end up with a lot of different mutated microsatellite alleles in the population. And that's what population geneticists take advantage of, those mutated microsatellite alleles. Thus, microsatellite mutations survive in populations, and alleles of microsatellites can be much more variable than alleles of genes. A microsatellite mutation might be an increase in length of the repeat number of base pairs or a decrease in length of the repeat number of base pairs. In a single species, the number of alleles will increase as the number of mutations in the population accumulates. Microsatellites can be used to determine whether a salmon that's caught by a fisherman comes from a particular river where that population is dangerously low. What's so amazing about salmon migration is that when the adults come back, they come back to the same river where they were born. The same river that their grandparents bred in, their parents, and that their offspring will also return to. And it's because the salmon come back to that same river that they have a unique genetic population. So each river has its own kind of unique genetic signature.
Now, back to the fin clip. Next is a quick step-by-step demonstration of how the fin clip is analyzed in the lab.
We take this little tiny bit of fin and we place it inside of a tube which contains lysis buffer that breaks the cell all apart so that the DNA comes free from the nucleus of the cell.
The next steps are as follows. Extract pure DNA. Load DNA onto plate. Assemble master mix of chemicals needed to amplify DNA. The master mix includes DNA polymerase, the enzyme essential for copying DNA, primers to select the segment for amplification, and nucleotides, the building blocks of the new DNA. Load plate with master mix. Place DNA and master mix into thermal cycler for PCR. Run PCR, polymerase chain reaction, to amplify DNA. The PCR procedure uses heating and cooling to amplify a small sample of DNA to millions and millions of copies for analysis. Use robot to transfer amplified DNA to fragment analyzer plate. Run amplified DNA through fragment analyzer.
The DNA travels through here, and then there's a laser that reads the color and also tells you the length of the size of the microsatellite as it passes past the laser. After samples are processed on the genetic analyzer, I use the computer to look at each individual fish's microsatellite signature, and then I use this information to determine that fish's river of origin.